Okay, good afternoon. We're going to go ahead and look to get started here, folks. So we have a panel discussion to get us started. And the panel discussion is potential use of eDNA for regulatory actions. Our first speaker up is Chris Logue. So Chris.
Yeah, hey, good afternoon, Dana. Thanks for asking me to be a part of the panel, and really appreciate the opportunity to talk with you today about eDNA. So, I wanted to start out just by talking about a couple of things that came up in the chat as a response or in the question and answers after we saw the previous presentation. A couple things that I think are really important to keep in mind here. We're looking at eDNA from the standpoint of, in particular, a detection tool. And in particular, we all, have been working on spotted lanternfly for a number of years here. And of course, we understand the fact that it's pretty cryptic and hard to find under low population situations, and we also know the limitations of the trap and the lure situation. So, our interest in eDNA really was sparked from some work that we had seen through our DEC on the aquatic side of things, and thinking about, how could this be used with a terrestrial invasive species, with the idea that we all probably have learned the lesson that I think that Julie started out her presentation with here a little while ago, of when you are finding things in the curve, the invasion curve is climbing rapidly. The sooner you find things, the more effective you can be with your resources. And there's a lot of work that supports that. So with respect to, I think there was a specific question about whether we would use eDNA, to call a county positive. I think that's probably a little bit premature. All of us on the regulatory side of things, we tend to be pretty conservative on these types of things. Also, I think, one of the things that we need to be wrestling with a little bit also is the potential legal issues associated with it. And again, I look at our plant pest law here in New York. Some of the parts of it date from the 1920s and 30s. And some of the tools that we use today were not even, a glimmer in anyone's eye when the rules were written. So that's kind of the way I'm thinking about this. I'm really thinking about eDNA from the standpoint of getting another good tool in the toolbox. Where do we go from here based upon, the work that Julie has done? For us, we have now some specific data points where her team has gotten some, what I would describe as fairly strong hits. I think I'd like to get our people out there and look in those specific locations. That kind of sort of pinpoints our survey a little bit and perhaps allows us to be a little bit more effective. So I think we're going to be doing some ground truthing on these data points and get a better sense of what they mean and where spotted lanternfly is. So we're not necessarily thinking of this at this point as a regulatory tool, but rather perhaps a better detection or an improved detection or survey tool that maybe can help us to direct our resources in a little bit more effective way. So a couple of other things, that I just would challenge the group with before I turn it over to one of the other members of the panel is some lessons learned. Those of you that know me and have worked with me, I hope you know that I'm always thinking out ahead and thinking about what are we learning from the data that we're getting. I think one of the things that really hits home for us And I will say that when I saw the eDNA map, I was not shocked by it, frankly. But I think we all need to be a little bit more skeptical of negative survey data. And I don't necessarily mean specifically on spotted lanternfly, but I'm thinking of negative survey data in general. And certainly in this case, I think in spotted lanternfly, that holds true. And then the other thing, I guess, that I would throw out to the group as sort of an open question is, are we serious, whether we're a State Department of Agriculture or a USDA or a county agency, whoever might be involved in invasive species management? Are we really serious about early detection and rapid response to invasive species? We can pick apart these results. We can probably find some cracks in them that maybe make us not want to consider them. But from my standpoint, I think I'm keenly aware of some of the limitations we have on survey. So if this is a tool that gets us in and makes us aware earlier that something is present, I think we need to really be serious about looking at it and following where the evidence takes us. I think perhaps it's a next step in the evolution of detection tools. There's probably nobody on the call here who's been around long enough to comment on my next point, which is I don't know where we were 50, 60, 70 years ago when pheromone traps became perhaps more common and were the cutting edge and a new thing. And so did we have these same kinds of questions about how we would use these tools? My sense is we probably did, because we were probably as a group more conservative at that time. So with that, I can pass it along to the next presenter or the next member of the panel. So thanks, Dana. And Stephen,
Thanks, yeah.
Go ahead, Stephen. Perhaps you would like to make some comments from the standpoint of National Plant Board.
Sure. Thank you, Chris, and thank you, Dana. First of all, that was a very enlightening presentation that Julie gave. Admittedly, I don't know a lot about eDNA and sampling, and really engaged in my first conversation with it earlier this week with the panel members as we were discussing how to create content for you guys today on this. But certainly it raises a lot of questions, many of which I think Julie acknowledges and Chris confirmed as well about how do we use it. And I guess I'm here today to represent the National Plant Board specifically on the topic. And I guess my message on this is that, with any new technology like this, we need to tread carefully, but I certainly see how this could be an important tool in the toolbox. And maybe in time, once we've vetted all of the potential false positives and ways that this could go wrong, then I think it could be even a tool that we use for confirmation of pest presence. I don't think I'm personally there yet. As I was preparing for this call today, I started writing down questions that I have, and I ended up with a page full of them. And the first question I wrote down was, can this be a regulatory tool? And I think that depends on the context of how you're defining a regulatory tool. Certainly, the way it sounds like New York is using it in finding what you believe to be a pest presence and then going out confirming on the ground, I would say we're already there. And thanks to New York's work on that already. But then to what extent do we want to go beyond that? Do we want to call that confirmed pest presence? And then I got to thinking about, okay, how could DNA spread? Wind movement. Certainly I'm down here in South Carolina and we get a few hurricanes and tropical storms every year. I would have concerns about whether DNA is spread that way and to what extent we could be potentially calling something a false positive. And then maybe with other pests, because I'm sure we're going to expand off of spotted lanternfly soon enough and talk about this for other pest detection. And you could have water movement as well. So still a lot of questions there for me. And then, of course, would states be looking to externally quarantine? I think these are conversations that we're going to need to have in the near future with the National Plant Board on how we're going to view this new data. Are we going to consider it something that an external quarantine could be issued over? Or are we going to try to reach an agreement where we're still going to look for confirmation on the ground again until we can answer a lot of those questions about how else could the DNA have gotten there? Or, would we just call it a regulatory incident until we have those boots on the ground confirmation? But, I guess my message here today is that it sounds like a fantastic tool. I think we need to be cognizant of the potential consequences to finding these positives and make sure we have conversations early on enough that we're all in enough agreement for what the information means to us, and then have an understanding of those false positives, and then again, just be very careful until we know those things. But I'm quite excited by the work that I saw Julie just present on. It feels like the sky's the limit, kind of like Chris, you just alluded to pheromones for trapping probably being the cutting edge. And I would imagine that had a lot of question marks around it when those first came along. But certainly I could see us moving past that in time with eDNA. And with that, I'll step aside and pitch it to another member as well.
Thanks, Stephen. Today we are going to have Dr. Kyle Beck from California. Kyle?
Yeah, thanks. So, yeah, thanks a lot. That was a great presentation. California, just to put it in perspective, California's perspective. So, it seems like a great tool for early detection. I'm not sure, like other people have alluded to, a regulatory action, from eDNA alone. I mean, there's a lot of issues with that, but certainly for targeting follow-up inspection, it seems like a really great tool. From California's perspective, it might be a little bit different, so spotted lanternfly isn't known to be present anywhere in the state. If there was if there was a positive eDNA hit anywhere in California, that it would be, maybe a little bit different, qualitatively than if you were in a state that was partially infested, so it'd be maybe more concerning or, demand much more urgent follow-up for that reason. And from a regulatory standpoint, California has always required, an identification for insects of an actual physical specimen and one that was collected officially. So, somebody just brings something into the lab, we're not going to take regulatory action on that. We'd have to follow it up with a find of a physical specimen. So obviously, at least from the way we are doing things now, and we've always done them, we'd need to find a specimen typically to do anything, to impose a quarantine or eradication. But certainly for focusing that inspection, and we're currently doing an annual high-risk survey for spotted lanternfly. We've got over 1,000 sites in the state. So, I'm just trying to think through, like, even if we took one sample from each site, just kind of pooled, I don't think it would be practical to have a separate sample from each tree. That would just be too much. But let's say we had one paint roller from each site. We've got 1,000 paint rollers to sequence. We do have a lab, can do molecular analysis, but it'd probably be I don't know what the workload, with over 1,000 of those in addition to what we do now, I don't know what that would cost. But, after hearing the talk today, it seems more, certainly I see it in a more positive light, or a more, I can definitely see the value of it, and we support, continued development of the protocol, and are definitely very interested in it. And I think I'm at the point where I'm just rambling on, so I'll pass it on to somebody else.
Thanks, Kyle. Appreciate that perspective. Scott from Illinois, Scott Schirmer.
Thanks, Dana, and thanks everybody. You know, I feel like I'm gonna echo everybody's comments as well, but you know, starting off with Stephen's comments from a regulatory specific perspective, you know, I think there are a lot of questions to ask and have answered, but I guess there's some solace in knowing that these are the kinds of decisions that we review and look at as a group. You know, if 50 of this or if 25 of the states said yes, we'll do regulatory actions, and 25 said no, we won't, we're not going to come to a consensus. We'll talk it through and kind of work together to get to a situation that everybody agrees upon. And I think that's going to be evident later on in a later presentation that you'll see. But you know, for those of you that know me, you probably know I like to use analogies quite a bit. And so I'm thinking about this as from like a fishing perspective. You go to a lake you don't know, you go around and you cast a lot and you cover a lot of ground, and then you kind of key in on where those fish might be. And so I kind of see this as a way to monitor, use that as an early detection tool, like Chris was saying, you know, maybe with sentinel plots or something, especially for a state that doesn't have it or doesn't know they have it, I should say. So you have some established locations where you may regularly monitor, regularly take samples and look for it. And then I think the other issue or other potential value from my perspective here is, and this may relate more to the situation in New York that Julie specified, is, you know, you have known infestations or known point infestations and monitoring the spread out from those. So you have your risk models that you use, your corridors, your pathways that you're regularly sampling to try and see, okay, we got it a mile out, we'll go 2 miles out, we'll go 3 miles out, and kind of monitor that spread and monitor not only where it's moving, but potentially the rate it's moving as well. So yeah, I agree with everybody that it's early, it's fascinating information. I'm glad Julie mentioned that most of what people know about this is from the aquatics applications, and that's kind of where I came into this originally, thinking with the Great Lakes here and the Asian carp and the monitoring going on in Lake Michigan. But it's fascinating and interesting information. I think it's a very applicable tool, and again, I think Stephen mentioned this too, is that being early, and Christopher as well, is that being early on, you know, breaking the ground on something like this. Who knows where it's going to be in 5-10 years? You think about these, you know, strips that we can use to test for viruses in the field. You know, what's the next application? What's the next piece of equipment that we might be able to use out in the field to use this quickly? So with that, I guess I'll wrap it up as well to save everybody from me rambling. And I believe Matt Travis with USDA is our last panel member on this. Matt?
Thanks, Scott. Appreciate it. And I certainly appreciate everybody's comments. A lot of wisdom and insight there. I'm going to start, you know, kind of with how Chris started it. Chris, we don't call them negatives, we call it pending positive, especially with spotted lanternfly. But I think it has potential. I think it looks like a great tool. It would be interesting to see how it could be expanded. Certainly, I would be interested in seeing how we could use this further, and really looking at how we review and vet this method and protocol. I think, to Kyle's point, a lot of agencies have a protocol and vetting process, a confirmation process that they go through either as a state or even as PPQ. We have our own protocol and process that we go through before we absolutely confirm the presence or positive results. And I see that as, you know, this is a beginning stages of a great new tool. It would be interesting to see how it would go forward, fully reviewed and vetted and you know, looked at by different agencies, how it could be used. I think it's an interesting perspective, and I agree too. I think with any new technology, it takes time to develop, and Julie's work is certainly very enlightening. It's very informative. A lot of interesting insights there, but I think that with any new tool, there needs to be some work done to make sure that, you know, when we do employ it, when we do use it, that we're doing so using the, you know, the best science and the best verification and review process as possible to make sure that it works in its one consistent standard for everybody who would use it. So I think that's important. I really appreciate the insight to my my other board members and panel members. It would be interesting also to compare it to, you know, some of the other techniques that we have, because we're always looking for things that are not only, you know, good scientific tools, but also better efficiencies. Like Chris kind of talked about, how can we make this more efficient, or is this more efficient than performing visual survey, or, you know, with egg masses we have canines now, and other aspects, other tools, and can this be another tool that we use? And is it more efficient? Does it save us time, cost, and effort? And I think a lot of those questions, at least for me, I see need to be answered, or I think that it needs to be answered on a larger scale as we look at, you know, Kyle's example of 1,000 samples. That sounds like a lot for a lab to process and go through and have a timely response in time to respond to, you know, a potential infestation if one exists. So I guess that's it for my part, and I guess I'll turn it back to you, Dana, and I guess I don't know if we'll open it to further discussion. Thank you.
Okay, thank you all. No, I think that this was a good perspective to hear. I think Julie's work, again, you know, it was originally developed for, you know, one use, and now we're considering taking it to a different level. How many other things are out there, so that we can, you know, again, we have to think outside the box. Spotted lanternfly doesn't fit into a neat pest situation that we've had before. So again, exploring all these different avenues are very good. So we are getting a few questions in the Q&A, and so Julie, I think this is really for you. What's the cost per the sample? Because that is going to have a large impact for some people.
Yeah, that's a great question. So it really depends on who you get to do the work. So California and really some other states are developing their own internal eDNA labs for a lot of the aquatic uses, because again, it's just becoming increasingly common for aquatic invasive species and also in freshwater and marine fisheries. So as those states have their own eDNA lab capabilities, then it's going to be cheaper to run it in-house for them than it is, say, to send it to my lab. And we're probably especially on the expensive side because I'm a research lab. So there are commercial labs out there as well that will run them for you. And those prices change. So I don't know that I can give you a flat answer, to be honest. It's not, once you have the genetic assay working, it's not that hard. It's sort of very unsexy molecular biology where you just run it through a qPCR machine. So
well, now, Julie, that all
huh?
depends on if you're a molecular biologist or not.
Oh,
Some
it's
people
true.
don't really
Well,
get into it, you know.
well, and I'm not really a molecular biologist. I'm more an invasion ecologist, and kind of, so it's like, oh, this is very useful. So anyway, it's, if I can do it, I guess the point there is, if I can figure out how to do it, anybody can. But it's not that expensive. And I showed you pictures of the field work, and it is very, very low tech. You do have to have a level of OCD. You know, there are protocols. And so we're kind of working at Rutgers and coming up with training materials also through New York State, but also for our own, because we get an increasing number of people asking, so that we can train people on how to do this well, so they're not doing contamination in the field and can go to scale. Like, there's no way, obviously, that one lab can support all this. It has to grow out. So that's, and then as it becomes more and more common, those prices will go down, honestly. So it's not, I will also say, like, the 1,000 one is high. Like, how many have we do stuff for, like, for shad and things for the state, New York, for New Jersey, and we'll do hundreds of them pretty easily. So 1,000 is not crazy, but it is high. And I think really if you're doing something for, like, California, you would use something like Matt's risk map, or something maybe that the state has already developed. to guide where you would go.
Right.
Because doing it randomly is not cost efficient. You
Right.
really
Yeah,
do
it's not,
need to
this
direct it.
isn't, yeah, this isn't a random survey, but yeah, it's a good point. I mean, we could use the risk maps to focus, you know, a higher high risk, you know, selection of sites, you know, from that larger, you know, number of sites and do, you know, sampling there. And also regarding the, you know, I wasn't really thinking about it as the assay, but that's also a good point. You know, you're not sequencing this stuff, you're just doing the bioassay. So that would be, it would be a lot simpler.
And Julie, I have another question for you. So the samples, once they're taken, the longevity of them, I mean, are they heat sensitive? If people are trying to ship, what, you know, handling, what kind of things are considered there?
You mean in terms of like once you got them on a roller or something? What do
Yeah.
you do?
Yep.
Yeah. So the good part about DNA is it does degrade pretty quickly, so that if you get a signal, you know, if you get a hit, you know it's happened. It's from a pretty recent, probably within a week presence of a SLF. The bad part is that once you collect that DNA onto your collecting device, roller, whatever it is, that you have to get it onto a filter pretty quickly. We've done
Okay.
some research showing that if you spray ethanol on the roller or you put it on ice, then you can probably keep the signal pretty strong for 24 hours. But we've thought about that in terms of just the operational use. Like you can't always have a truck with a filter thing on it, you know?
Right.
So, and I think the limit we've seen is like, you probably need to get it to the point where you can filter it within 24 hours. Once it's on a filter, it's fine because it says these little Eppendorf tubes with ethanol, and they'll save forever.
Okay. Good to know. Yeah, that would be another consideration, especially for the larger states trying to get samples to a lab. So yeah. So Stephen, I thought that you were making the statements very well, and I just wonder, you know, from the National Plant Board perspective, given that we are seeing this, is this something that you're going to consider to move forward and encourage, at least on a regional level, people to start having these conversations?
I really do, Dana. You know, like I said, I really just started getting up to speed on this topic in the past week or two. I didn't realize that New York was already doing all this work, but it seems like we're already a little behind. But certainly, I think I would encourage the regional plant boards to begin discussing it at their regional meetings in the next few weeks to a few months, and then if there's still room in our agenda as we're planning that for the annual meeting in August. Maybe we can still work something in there to have a conversation about it, but absolutely, I believe that it's imperative that we have an early conversation and try to get on the same page. And I know depending on the state, we're limited in how much control we have over our regulations at times. But certainly, if we're going to have an impact on that, I think it's important that we have this conversation earlier. And Dana, on a bit of a related note, I actually wanted to ask a question of Kyle. Kyle, you mentioned that in California, you would require the actual pest being present, such as an insect sample. And quite honestly, we'd be very similar to that in South Carolina. But to play a bit of devil's advocate, I got to wondering, does doing molecular analysis on nematodes move into that same territory where you're really just dealing with DNA at that point? Certainly, that's a pest that you can't see with the naked eye. You can see it molecularly, but then it gets complicated. Then I just wonder if there are comparisons there, or
I'm not sure that I'm not a nematologist, but I know if you were sequencing an insect, you would have the specimen. Typically you'd have the specimen, and then you crush up a leg or something like that, and also typically you would first look at the morphology, and then you might use the molecular data, to support the identification. But I don't know how nematologists do their sampling, but I'm assuming that they aren't taking an environmental DNA sample of the internal contents of you know, some insect or something like that, you know, that they're actually taking nematodes and they can visually, you know, confirm that they have specimens, you know, and then they sequence that.
Mhm.
I will say in Pennsylvania, that's what we require, even with nematodes. You know, we consider that a pathogen. So we still require a physical sample that is then confirmed in our labs with traditional and molecular.
Okay. Yeah, I thought I would toss it out there, but you know, it's hard not to see that we're probably all going to be undergoing some regulation changes, especially if this technology develops the way it's sort of looking like it would. I think we're all going to need to establish definitions within our regs, and then you know, account for that ability, or at least spell out how we're going to allow it to be used and not be used in pest detection positives and such.
Now,
Yeah, I can add to that quickly, and I think there's two things to put on your radar. Is one is the same discussion is happening within aquatic invasive species responses, and so it's a broad discussion about how to, like, this regulatory discussion is common, and no one is past where you guys are in terms of deciding how to use eDNA in a regulatory context. And the other is that in Australia, for example, they're using this a lot, and they're moving towards a certification process so that labs become certified eDNA labs, and they pass muster, whatever that is, you know. So that's a way that I suspect that's where we're going to end up moving for eDNA, period, for all the different uses, because it does transcend even invasive species, is that labs will have to go through some sort of certification process. So those are things to kind of keep on your radar.
Julie, I appreciate that, and those are good points. And I think really what this is, the bigger picture here is that we've got to make sure that research and regulatory are having conversations on a regular basis, not just when it comes to a certain pest, so that we are kind of aware of what you guys are using scientifically to diagnose and identify, so that we can try and stay current as we possibly can with our regs and our ability to utilize the science that you develop. So I'm hoping that that is something that we will take away from this, from the information you've shared and from the concerns that we're hearing from regulatory and things that we need to consider, because for some areas, it takes a long time to move regulatory into a certain direction. So we need to keep that in mind. Other questions, other comments from the panel? Julie, I will let you know that there are some questions that I think are for you, really, regarding the eDNA. So if you don't mind taking a look at those to see if you can answer those, that would be great.
Yeah, sure. Do you want me to do that quickly now, or?
I think we've got time. If there's no one else from the panel that would like to speak up, we'll go through those. So one of the questions is, could samples in areas of low likelihood be batched to lower cost?
Absolutely. That's an excellent question, and that's one thing we're waiting to hear for some Forest Service money to see whether or not we can explore that with Asian longhorn beetle. And it's just like trying to hit the sweet spot of maintaining a high detection threshold, but batching as much as possible.
Great. And another one is, are you going to spread out your sampling points for eDNA next year? It seemed that the points were very close together this year.
Well, yeah, we might do that. Yeah, we have to talk to our working group out in New York State, and we're going to, after our results this year, we thought we were, you know, on the front edge, and we clearly were not. So we're going to talk with them about how to spread out, getting lots of good feedback from this summit on what would be useful to explore. So we'll spread them out for sure. And we're at least going west and north, and we may spread them out in between each one. this was sort of the first time we tried it. So that seemed reasonable.
Okay. And do you think that this is something that can potentially be ran as a portable PCR in the field?
Yeah, that's another great question. And the answer is yes. So we're going to use, actually, I think we're going to use some of our SCRI money that from working with Julie's group and the last bets and get a portable qPCR that's capable of running, that we'll figure out, do all the things we need to do so it can run the assay that we have in the field. So you could imagine in that case that, you know, 2 people in a truck with the right tools can do on-site eDNA detections with immediate results. It can't do large batches, right? But the idea of having it be more of a strike team is interesting. But yes, absolutely.
Great. Thank you. And actually, I think the next one is more for Julie Urban, because of your work in genetics, Julie. Is anyone currently investigating an application of CRISPR technology to develop a lethal gene specific to SLF that would over several generations reduce or possibly eliminate SLF in North America?
There's a guy, Jason Ragston, who's a researcher at Penn State who is kind of a CRISPR expert. And so, yes, he hasn't really started on it yet, but he and I have talked about doing it. And I know Rick Roush has kind of encouraged that a lot. And so, yes, that is something that he's interested in particular.
Great.
I also want to point out there's kind of a lively conversation that we're having in the chat.
I saw that.
Because again, thinking, you know, one of these issues when I was asking Julie when these samples were collected and thinking about, I was shocked to see the throughway, all of those detections up to Albany. And so the idea of using the eDNA sampling to potentially delimit what could be the range of their northern limits in a sense, right? And so the other question though is when you're talking about samples collected like in October, when they're flying, they're getting into stuff, you know, for this northern limit idea, they could be they could be in areas feeding heavily and excreting, but those areas still might not be conducive enough for them to be able to establish a population in terms of temperature limits. And so I think though that would be neat to then figure out where exactly, what is that range you need to look in that could be the northern limits to then look for subsequent life stages to know that populations really were persisting there. So that's kind of what my take on all that.
Yeah, and I think that that's a really interesting perspective. And I just wonder, because I know that one of the Penn State folks have been doing having people report when they're seeing birds feed on these. And I think we're getting more and more reports of that happening. So now I'm wondering with the eDNA and birds eating, is that going to cause confusion? So I don't know if that aspect has been considered, looked at, in what that would do. So I think it's very interesting. Anything else? Any other questions? Let me double check here.
Dana, in the chat, Stephen mentioned something about evaluating biocontrols and then potentially other treatment evaluations by using eDNA in an area that's been treated or received. So there are,
Yep.
Seems like every second there's other ideas and other novel potential applications for it. So
I know I don't think we realized how hot this topic was going to be when we chose it, but yeah, there's a lot going on in the chat. So I'm glad that it's going to be captured. So yes, and so we have another question. Is eDNA a yes or no tool? Are trapping visual surveys important for established population density? And my takeaway, and Scott, I'd love to hear your perspective, is eDNA is a tool, absolutely, to put in with trapping and visual surveys. And I think though that it pairs with what we learned about from Matt is, you have to take all these tools. So you take the tools that Matt has given us through Temple to say, okay, where is the greatest possibility that this could happen? And then you start utilizing the visual surveys and the eDNA at that point. Would you agree or not, Scott?
No, I would totally agree, Dana. And that was kind of my point behind the Sentinel plot comment, where we have a rail yard in Chicago that has Tree of Heaven. That's where we go every week or every month to take a sample. So yeah, I think it'll give you that kind of ability to pair and partner up with all the other tools in the toolbox, and we'll keep saying that. But yeah, using the risk models, again, I made the comment about casting a wide net, but again, that I didn't mean that as randomly checking trees for this. We all have to hedge our bets and figure out, use the models, use the data, the information that we have to hone in where it's most likely going to appear. And of course, what we found is, or what we've seen is that that's not always necessarily the case. There's going to be curveballs. Just look at Indiana, for example. So,
Right.
But I couldn't agree more, Dana, is that, there's 3 towns in Illinois where I'm expecting it to pop up, and fingers crossed it pops up there and not somewhere that really crosses your eyes, so to speak. But yeah, I think that's the application that I would see most applicable here under our current situation. Yep.
Great. So, well, everyone, thank you very much. This, like I said, I think the eDNA topic is one that we're just starting to explore, and I look forward to learning more as it develops and learning more about how regulators are going to be incorporating those tools into what they're currently doing for spotted lanternfly as well as any other invasive pests that we may come across. We are right at close to the 1 o'clock mark. So I'm going to say let's go ahead and break for lunch. We do get an hour for lunch, so it'll be 2 o'clock Eastern to come back. But again, thank you all very much to the presenters this morning, and to our, again, to everybody who's behind the scenes, Jacob and Brian and just everybody. You're doing a fantastic job. Our numbers, again, are exceeding what we saw last year. So thank you all for participating, and thank you for the interest. And we'll be back here in an hour. Thank you.